Okay, so this video is a review of Unit 4, which makes up about 10 to 15% of the AP Bio test, and it's on cell communication and the cell cycle. All right. So this is just a statement that says variation at the molecular level provides organisms with the ability to respond to a variety of environmental stimuli. So in this unit, we're going to look at how cells can communicate with each other in response to environmental stimuli. And that variation in the numbers and types of molecules within cells provides organisms a greater ability to survive and or reproduce in different environments. So those variety of number and types of molecules could be referencing like hormones or different types of receptors that make them or allow them to survive and reproduce, like have high fitness, have low fitness, etc. So first, cells communicate by cell-to-cell contact. Now, one of the best examples, I think, of cell-to-cell contact would be part of the immune system, where you have cells come in direct contact with each other, but you also have like your helper T cells in direct contact with like your macrophages or your antigen-presenting cells. We also have example in plants of plasmodesmata, which is like the little channels between two plant cells where messages can travel from one cytoplasm to the next. So that's cell-to-cell contact. But we also have cells can communicate through short distances, which would be making use of local regulators. So again, other examples of short distance local regulators would be found in the immune system. So helper T cells release cytokines that can activate both B cells and T cells. There's also in your inflammatory response, there's chemokines and histamine that are released in your second line of defense that only travel a short distance. But we could also talk about things like growth factors, like platelet-derived growth factor that only travels a short distance. You could talk about neurotransmitters in the synapse between two neurons would be a local regulator, as well as the pheromones that bacteria send out in quorum sensing would also be an example of a local regulator. So with short distance or local regulators, those chemical signals aren't going to travel like in a bloodstream. They're going to travel a very short distance to cells in the vicinity or to cells that are nearby. Then we have long distance signaling, and long distance signalings will be your hormones that are produced by a gland or an endocrine cell or an endocrine gland, and then they'll travel in the bloodstream to their targets. And when we talk about a hormone reaching its target cell, like what does that mean, right? So a target cell is a cell that has a specific receptor for that hormone. Now, the receptor can be a cell membrane receptor, like a plasma membrane receptor, or it can be a receptor for a lipid-based hormone located within the cytoplasm. So when we talk about hormones that are like a peptide hormone or a protein-based hormone that are hydrophilic, they cannot diffuse through the membrane. So therefore, they would, like in this example here, attach to a receptor that's bound within the membrane to activate a signal transduction pathway that ultimately brings about a cell response. So when we talk about these signal transduction pathways, though, and we talk about like protein modifications, one example here is you can see how this GDP protein becomes active to GTP. That's a protein modification. But you also see these tyrosine kinases, these orange proteins represent receptor tyrosine kinase. And initially, a receptor tyrosine kinase is two like unphosphorylated, polypeptides. And then when the ligand attaches, like it forms a dimer. So that is modified, but also to pass the message on from that tyrosine kinase, it also relies on relay proteins. So like relay proteins would be like this yellow one here that can like relay the message. And then a phosphorylation cascade, when we talk about kinases, it's an ACE, which means an enzyme, but a kinase transfers phosphate groups to proteins. Like when we talk about a protein becoming active, oftentimes it's because it was phosphorylated. Like a phosphate group was added, and then that protein will go through a conformational change, which means a change in shape, and become active. So if you look at this diagram, inactive protein kinase 1, when it's phosphorylated, you can see a slight change in shape, and now it's an active kinase and can phosphorylate the next protein kinase in the cascade. Now, one thing I did not do a good job in my classroom of doing is emphasizing that in these cascades, though, the signal like amplifies within the cell. And so, anyway, many signal transduction pathways, same standard. So here, like I believe this picture is from Khan Academy. If epinephrine attaches to a receptor, here you have adenylyl cyclase will become active. And then adenylyl cyclase will convert ATP into cyclic AMP, which we use as our second messenger. And here you can see how that will activate a phosphorylation cascade. Then you can see at the very end of the cascade, you have a phosphorylated protein that now is active and can hydrolyze glycogen into glucose to raise your blood sugar. Versus over here, if you are in the fight or flight response, which is when epinephrine is released, you are going to need glucose. You're going to need energy to fight off whatever's happening or flight. And so therefore, you're not going to store glucose as a polysaccharide. So therefore, you can inactivate the enzyme that would be used to make glycogen. So another thing I want to point out when we talk about phosphorylation cascades is that when you have a peptide hormone, a protein-based hormone like a number 1, which is a signaling molecule or a ligand, and it attaches to a membrane protein, activating a phosphorylation cascade, these cascades, they rely on the use of a second messenger, which is number 7 right here, because otherwise the signal would stop at number 2 right here. And so because number 1 cannot cross the membrane by diffusion, it relies on number 7 to pass on the message. Okay. So we have different types of receptor proteins. And so when we talk about receptor proteins, a G protein coupled receptors are an example of a membrane-bound receptor protein. They rely on a G protein to pass the message or relay the message from the receptor to activate the proteins within the cell to start that phosphorylation cascade, or that second step called transduction. Now here are tyrosine kinases. I want to point out though, like when we look at a tyrosine kinase receptor as well as a G protein, so it counts same thing for a G protein linked receptor as well as an ligand-gated ion channel, as well as a tyrosine kinase. I didn't save it. But there are different regions. So the part of the protein that is outside of the cell is called the extracellular domain, or like the extracellular region, extracellular meaning outside of the cell. And then you have your intracellular region. So right here where it says the intracellular region of the protein receptor will change its shape, initiating that signal or the transduction signal. So here, when we look at, let me move my face. When we look at this receptor, tyrosine kinase, once the ligand attaches, right here you're going to see some, like this would be like a relay protein passing the message on. And then this IP3 is another example of a second messenger, similar to cyclic AMP. Okay, so then you have this area right here is the part of the receptor that is located within, like it spans the membrane. So that's going to be like your transmembrane region of that protein receptor. So again, this is the extracellular domain outside of the cell. Here's your intracellular domain. And then the part that is right here is the transmembrane region. And if a mutation happens in any of these parts, it could impact the cell signaling ability of that cell. Okay, so I liked this picture because of this second messenger that's not cyclic AMP, to kind of just show that there's there's more than just cyclic AMP that will relay or amplify the signal within the cell. Okay. We also have ligand-gated ion channels, and this is common in your with your neurons, or like your neurotransmitters would be the. So in our nervous system, the neurotransmitter that's released from one neuron will act as the ligand on the next neuron, the postsynaptic neuron, that will then attach to the ligand-gated ion channel, which will then cause the ion channel to open and ions can flow in, which will lead to hopefully or hopefully not, either way, or possibly, I guess you should say, the postsynaptic neuron meeting its threshold and leading to depolarization and the action potential, unless, of course, it's an inhibitory neurotransmitter. But anyway, there's a whole action potential video on that subject. So those would be your ligand-gated ion channels that open the receptor or open the protein for ions to flow into the cell. Now, signal transduction pathways, they do influence how a cell responds to its environment. And so here you have your growth factor, which is your ligand attaching. These would be your, looks like tyrosine kinases. And then how a cell responds to its environment, you can see here, a growth factor is like its name for growth and cell division. And so the ultimate cell response is cell growth and division. But there's a lot of steps in mitosis occurring. So here you have one of the things is to turn on like a transcription factor, to turn on the genes that are necessary to produce the proteins that lead to cell division. So a ligand attaches that causes a whole chain of events within the cell to bring about cell responses. Now, sometimes the cell, it could be like for the cell cycle and mitosis for growth and repair. But sometimes it can also cause apoptosis, which is programmed cell death. There are times in our development where it's crucial that a cell is signaled to self-destruct. For example, like the webbing between our fingers as we're a developing fetus, or sometimes if a cell is just too damaged. If the DNA is too damaged and it's an unhealthy cell, we don't want that going through the cell cycle, so the cell response would be apoptosis. Now, it's also important to realize that some mutations in any domain. So earlier when I was talking about the different domains of a receptor protein, so if it's like a mutation in the receptor region or in the like the intracellular part, could cause malfunctions in our cells, and so they can affect any part of it. So one example in a relay protein called RAS is sometimes, let's pretend that a growth factor, GF, is the normal ligand for this receptor, and ideally it would cause the cell to divide. However, sometimes this relay protein can be mutated and it stays active. So even in the absence of growth factors, this cascade stays on, leading to uncontrolled cell growth. So there can also be chemicals that interfere with any component of a signaling pathway and that may activate or inhibit the pathway. So, oh, I don't have a picture, but when we take certain medications, let's pretend a person is maybe underproducing a certain hormone. Well, then you can take synthetic or man-made hormones that mimic the ligand activating the pathway. Or on the opposite, sometimes a medication or a drug could act as think about like a competitive inhibitor that blocks the ligand from attaching and therefore stopping the cell response. So when we talk about activate or inhibit, it would be like molecular mimics, that have the same chemical properties that can attach to that receptor, causing either increased signal transduction pathways or decreased. And that's cool that scientists have figured that out because we can use it in a lot of medical situations. All right, we're going to talk about feedback. So organisms use feedback mechanisms to respond to internal and external environment changes. So in this one would be like body temperature, your body like you sweat, and your metabolism slows down on hot days to bring you back to homeostasis if your body's overheating. If your body's too cold, then your muscles will shiver and your blood vessels will constrict in order to bring your temperature back up. But we also will use hormones and cell communication in order to maintain homeostasis. So a good example is insulin and glucagon. They're both ligands that will attach to receptors that will help the body to respond to maintain a constant blood sugar. So after a meal, if your blood sugar is high, your pancreas will release insulin, and insulin's receptor is a tyrosine kinase. And the cell response is to increase facilitated diffusion to allow glucose to enter into the cell to leave your blood and be stored in the cell of like your liver or your muscle, so that way your blood sugar balances out. You'll store glycogen, but on the opposite, if you haven't eaten, then the hormone glucagon will be released, and the cell response will be to hydrolyze or break down glycogen and then increase your blood sugar. So our body uses cell communication to help maintain balance. We also see this in like osmoregulation. In Unit 2, I talked about osmoregulation in organisms to maintain water balance. So we will use the hormone, antidiuretic hormone, from our posterior pituitary. We'll release that and it'll communicate with our kidneys to help maintain proper water balance so our cells stay isotonic. We also, so like here you have like a calcium balance to maintain homeostasis. So my advice to you, if you are studying this standard, is to be familiar with how to read feedback loops. The idea in negative feedback is to bring you back to set point. And the body will use hormones most of the time to keep us in balance. And those hormones are ligands, and that's why it fits all together in Unit 4. Now, sometimes the hormones, though, can be steroid-based hormones like testosterone or estrogen, and their receptors will be located within the cell, and then they'll directly act as a transcription factor. So kind of skipping that phosphorylation cascade part. Now, what I was saying, though, is if you're studying this, College Board most of the time will give you the names of hormones, will give you diagrams of feedback loops. It's more that you're able to read and understand it, especially hormones that you're not familiar with. They like to give you like a new idea or concept and see if you can take what you already understand and apply it to a new situation. Now, in positive feedback, though, positive feedback is going to amplify the response. And so like childbirth, the contractions get stronger and stronger and stronger, or in blood clotting, a blood clot that forms in the walls of a blood vessel will amplify, like get larger and larger and larger, or like ripening in fruit or lactation and breastfeeding in mammals. So in positive feedback, the stimulus results in like an amplification, which causes more stimulus and more amplification. And so that would be a positive feedback loop. Okay, so now moving on to cell division. In eukaryotes, cells divide and transmit genetic information via two highly regulated processes. So you notice this is eukaryotes. We have both mitosis for our body cells, our somatic cells, and we have meiosis that will make our sex cells or our gametes. What you don't see here is binary fission because that's in prokaryotes or bacteria and archaeans. So when we talk about it being highly regulated, we have what's called the cell cycle. And the cell cycle has a couple different parts. You have your interphase that is made of G1, S, and G2. In G1, your cell is living life and existing. And then an S phase, like if a cell receives a signal to divide, at that G1 checkpoint and it enters into the S phase, the cell is going to divide at this point. So in S phase, your DNA goes through DNA replication, which uses the enzymes DNA helicase, DNA polymerase, etc. And it's going to make like sister chromatids. So here's your linear chromosome, and then it's going to duplicate and form your sister chromatids that are identical copies of DNA that happens in S phase. So the amount of DNA doubles in S phase, and the cell prepares in G2 for actual division, and then that is the end of interphase. So interphase is G1S and G2, and then M phase is made of mitosis and cytokinesis, where the cell will actually divide the nucleus and the cytoplasm. Now, if a cell is non-dividing, like a brain cell or a liver cell, it will enter into a cell cycle arrest where it just stays in G0 or like G1, like forever. Or sometimes it might receive a signal to actually divide, but a non-dividing state is the G0 or G0 phase. Okay. So mitosis is a process that ensures the transfer of a complete genome. So with prophase, metaphase, anaphase, and telophase, the idea here, this is for growth and repair, and also asexual reproduction. And at the end of mitosis, you'll have two completely identical daughter cells. And yeah, if you want to study in more detail what's happening in each step, I would watch my videos or any video on mitosis on that to be familiar with the vocabulary like spindle fibers, centromere, sister chromatids, kinetochore, metaphase plate. Like I would recommend becoming familiar with each step in mitosis. Okay. And then there are checkpoints, though, in the cell cycle to regulate this cell division to ensure that our cell cycle divides only healthy cells, right? So at the G1 checkpoint, that is to ensure that the cell's DNA is healthy and intact. So we're only replicating healthy DNA and healthy cells. Then at the checkpoint, like during S phase, before you go on to G2, you want to make sure that all the DNA has duplicated and it's all in the form of sister chromatids. And then in M phase, or before M, there's like a G2M checkpoint to make sure that the cell is like all ready to divide. And then during M phase, there's a checkpoint to make sure all of the spindle fibers have actually attached to the centromeres or the kinetochores, so that way when they do pull the sister chromatids apart, each daughter cell has an exact copy of the DNA. So there are cell cycle checkpoints that ensure that the cell is progressing correctly through the cell cycle. Now, within each step of the interphase and cell cycle, though, to regulate these, we have cyclins and cyclin-dependent kinases. So cyclin-dependent kinases stay constant in amount in the cell cycle, but they're only active when there is cyclin. So the amount of cyclin varies during the cell cycle, and together, when cyclin attaches to cyclin-dependent kinase, like we see here, it is now active, and we saw earlier in the video a kinase phosphorylates proteins. So when it adds these phosphate groups, these are now like activated proteins. And so here in the G1 to S checkpoint, if we're saying yes, divide, it makes sense to activate this CDK so these target proteins can actually begin replicating DNA. So to make copies of your DNA relies on enzymes so that replication process can begin. Now, disruptions to the cell cycle may result in cancer and or programmed cell death or apoptosis. So if there is DNA damage in a cell, you don't want that cell to divide. That would be harmful. So we actually have this molecule called P53, and P53 will kind of like halt or stop the cell from dividing and passing that G1 checkpoint. So normal P53 is good at protecting us from developing like cancer because it stops and it will like halt, like create like a CDK inhibitor that will stop the cell cycle from dividing. So here, if P53 binds to DNA and the cell response is produce this inhibitor, it'll stop the cell from progressing through that checkpoint and either allow the DNA to get repaired or trigger apoptosis. And then if it's all fixed, then it can go through the cell cycle. However, in 50% of human cancers, there's a mutation and P53 does not work as planned. And you can see that there's no inhibitor. So that cyclin-dependent kinase gets activated and you have like damaged DNA getting copied as it goes through the cell cycle. So P53 is super important at regulating whether or not the cell passes that G1 checkpoint. Oh, and I guess this is the end of Unit 4. I do have videos on each of these topics in more detail if this was too quick of an overview for you. So good luck on your AP test or your unit test, whatever you're watching this video for. Good job, guys.